It's my great pleasure to introduce our speaker for today, Michiel Vandenbosch, who, as I mentioned, joins us from the M4i facility in Maastricht. And M4i stands for the Maastricht MultiModal Molecular Imaging Institute, which is a core facility focused on mass spec imaging. Michiel has got his PhD in pharmacy from the University in Leuven, where he focused on forensic toxicology. And already at that time, he was partially working, in part working with mass spectrometry, and that must have stuck because then during his postdoc, he went to Maastricht University focusing on mass spec imaging to study lipidomics and Parkinson's disease. And he, since 2020, is a core facility manager at the M4i Institute, and also leads his own research group that focuses on the evaluation of new therapeutics in a multi-omics approach using mass spec imaging. I'm really happy that Michiel can join us today and present the facility and the work that's supported by the Maastricht node of Euro-BioImaging. Michiel, please take it away.
Thanks for the invitation, first of all. So I don't have to introduce myself anymore, but I'm the core facility manager at Maastricht MultiModal Molecular Imaging Institute. Now, when we talk about mass spec imaging, people always say it's just pretty pictures. Now, hopefully, during this presentation, I can convince you that what we're doing is making more than just pretty pictures. Now, an overview of this presentation. First, I'm gonna tell you something about where we're located in the Euro-BioImaging field. Then a short introduction, what's M4i, what are we actually doing here, and what do we stand for? I'm gonna start very, very basic about mass spec and what's mass spec imaging, to dive into some of our projects, to show you of what we're actually capable of doing, and then how we hope that in the end, this all translates into the breaking the boundaries to meet clinical needs. So let's start off. We're part of the ME node here in Maastricht. We're a mixed node, so we offer a lot of techniques all in the same place. We have different groups participating in this node. One is with Professor Mark van Zandvoort, who's more into the advanced optical microscopy. We also have a nanoscopy core facility. They offer everything going from cryo-EM to SEM to TEM. There's an entire list of techniques that can be used. And then we also have Professor Ludwig Dubois, who offers micro-CT, PET, SPECT, and so on. And we're located actually in the middle of Europe, all the way in the south of the Netherlands. So very reachable for all of you, I guess. And then we go to our core facility. Who are we? We're part of that node. And we are the M4i mass spec core facility. We offer collaborations to everyone. That's why it's open. We are in a research environment, but we're also linked to a university. So we also participate in education and all the academic tracks that are accompanying that. We offer services. So if you have samples that you just want to be analyzed, you can send this to us, and we can send you a quote and in the end an invoice and so on. But we also offer that in a collaborative way. And we also offer training on all the mass spec instruments we have. We are one of the biggest mass spec institutes in Europe for sure, and maybe even in the world. And what we offer are mass determinations, everything mass spec related actually. So we look to drugs, proteomics, lipidomics, metabolomics, polymer analysis, and so on. But we have a specialization in looking at the spatial distribution of that all. Now, I run the core facility, but I have, of course, have some lab technicians helping me, so I don't do this work all alone. But the cool thing about M4i is that we're actually embedded in a research infrastructure. We're part of the university. Our director, Professor Ron Heren, who leads M4i, he's very well known in the imaging world. And our institute is divided into two divisions, one being nanoscopy, where we more look into the cryo-EM and so on. And then we have our division of imaging mass spectrometry, where also our core facility is embedded. And in this research infrastructure, every PI has kind of its own specialization. Some are more into building actually the imaging instruments, while others are into bioinformatics, trying to combine all the imaging modalities that we have, while others are more in the application field and try to find intraoperative ways for diagnosing and imaging. So 8 PIs with all their own specialization, but in the end, our mission is all the same. We try to perform fundamental instrumentation and applied studies as a part of a translational, synergistic, interdisciplinary research program. We're unique in that kind that we're one of the leading players in the molecular imaging field. And we also have top-notch infrastructure, if I'm allowed to say that. So the cool thing is we are linked to the hospital too. So what we try to do is cover all the scales all the way from the cell to the patient. Cell, we have our nanoscopy department where we can do cryo-EM and so on. Then in the middle, we have mass spectrometry. So we can tackle all the way from the nanometer scale down to millimeters. But we're close to the hospital, so we also have access to CT scans and so on to look at a broader picture. Now, what's mass spectrometry? Well, in mass spectrometry, we have the capability to measure masses of individual molecules that have been electrically charged. In our experiments, we very often have thousands of compounds in a single experiment, and we try to identify the unknown compounds. Now, how do we do this? When we go back all the way to our chemistry classes in high school, we all remember this table, which is a periodic table. When we have a molecule like CH4, for example, we can look into that periodic table and look at the mass of the carbon element and the hydrogen element. When we combine this, we get a molecular weight. We get an identity of this molecule. We don't measure this using a normal scale. They are not sensitive enough. No, we have our mass spectrometers, and one of the most common used mass spectrometers is a TUF analyzer. Very simple. We have a long tube where we introduce our sample on the left. We're gonna charge the molecules, give them an electrical charge, and then we're gonna accelerate them in the tube. And I always compare it to the same as with humans. If you would put them all on the beginning of a street and you let them run towards the end of the street, very often the small, tiny, light-weighted molecules, humans, will reach our detector first, while the bigger ones that are very often a little bit slower. If we then measure the time that it takes to run through this tube and reach our detector, we can put a number on it. We can differentiate between the masses of molecules. So in theory, the workflow is almost always the same. We have a sample that we introduce. We give it a charge, and then we're gonna let it run through the mass analyzer. And when it reaches the detector, we do the data processing to identify our molecule, or at least the mass of the molecule. We are looking at a special kind of mass spectrometry here. We are looking into mass spectrometry imaging. And this is a label-free technique that enables us to detect, localize, and identify molecules in very different and also complex biological samples. We have the capability of looking at lipids, proteins, metabolites, polymers, or pharmaceutical compounds. Now, modern MSI techniques, there are a few, but today we will be mainly focusing on multi-MSI. Also, I'm going to give a short introduction into SIMS and DESI, because those are also techniques that are available here. When we look at MALDI, it spans the range all the way from proteins down to small metabolites. DESI is more in the middle, where we have from peptides to metabolites, while SIMS is more specialized in looking at elements. If we look at the spatial resolution, so what's the smallest detail we can see, we see that in SIMS, we can go down to 100 nanometers, while MALDI at the moment we're limited to, we're pushing it forward, but let's say 1 to 2 micrometers. DESI is a little bit less good in spatial resolution. There we're stuck at hundreds or 50 micrometers. But if we look at the time it takes to run a sample, for DESI, we don't need sample prep. We'll discuss later. It's quite fast. MALDI needs some sample prep and then some analysis time. But SIMS takes a very long time. So it's always a trade-off between which molecules are you interested in, what's the spatial resolution you're interested in, and how fast do you want it to go. If we now go into SIMS, or secondary ion mass spectrometry, in this technique, we're actually gonna have our sample, and in our imaging field, we most of the time work on sections of sample. So we make a section of our sample, put it on a microscopic slide, then we put it in the analyzer. With SIMS, we're gonna bombard our sample using an ion beam. So on the left side, you see an ion beam reaching our sample, and the energy is transferred from the ion beam to our surface, and we create ions in this case. So we sputter away the sample surface with a lot of energy. So in SIMS, we have intense fragmentation, which makes it hard to look at the intact molecules, but we have a bigger chance of looking at elements. So if you're interested in looking at gold or sodium or silicium on a surface, SIMS is the way to go. The cool thing about SIMS is you can make a surface image, but you can also make a depth profile. Then we'll sputter away the surface and create like a hole. So we're gonna go in depth, so we can create a depth profile image of our sample. In DESI, or desorption electrospray ionization, instead of using an ion beam, what we're doing is we're actually moving an easy source, so an electron spray source, over our sample. So very similar to what we do with LCMS. We have our capillary that's heating a solvent, and that solvent is being sprayed on our tissue. So this easy source will move over our tissue, spray the electrospray droplets, and then as a result, ions will be created and moved towards the mass spectrometer. Now, what we will mainly be discussing today is MALDI imaging. In MALDI imaging, we also make sections of our tissue, of our sample, and we're gonna cover it with a matrix. Our matrix, actually the only goal of the matrix is to extract the analyte we're interested in from the tissue into that layer. And when we hit it with the laser, it has to help ionization. So we cover our tissue with matrix, and in the next step, our laser beam is gonna shoot at a specific point, and the laser energy will be transferred to our tissue, will create ions, and those ions will be transferred to the mass spectrometer. Now, if we do this point by point, so first we shoot left top, then we move a little bit, shoot again. If we then recalculate the position of where we found the ions, we can create these heat maps that you see on the right. Very similar to in sports. We can then see where a molecule is highly concentrated or where it's less concentrated. And in the next step, we can use this to draw conclusions regarding the location of the molecules. If we look at this in practice, I made a short video that should start. Okay. Like this. So here we see our section of our brain. We selected the region that we want to image. On the left side, you also see some spots. When we know what we're looking for, we very often put standards on our slide, and then we're gonna let our laser run over it. And each time it shoots, it blinks, we can create a mass spectrum. So you see a full mass spectrum of all the ions that were extracted from that region. In the next step, we're gonna throw it in our software. And in that software, we can see an average spectrum. So a combination of all the spectra that were collected. The right side, you see all the samples. These are all the samples, just optical scans to have an idea of how it looks like. Here we see the average spectrum, and when we zoom in on that average spectrum, we can see a peak. This is an ion that was located in the tissue. When we select that ion, we can create the ion images. And if you then zoom in, what we can do is play a bit with the intensity of the scale, because it's all semi-quantitative. And if you zoom in, you can see 1 pixel in this case was 30 micrometers. So the laser shot every 30 micrometers. So we had molecular info of each 30 micrometers of the tissue. Now, in our institute, I always say we have more mass spectrometers than people. So we have an entire range of mass spectrometers, and each of these mass spectrometers have their own capabilities. Some of them are really fast, but when something is really fast, very often it's not that accurate. So when we want a more accurate instrument, we have to pick another one. Then in the next step, sometimes an instrument doesn't need to be that accurate nor that fast, but we want to make, for example, a depth profile. Or some instruments need conductivity while others don't. So we offer an entire range of mass spectrometers with all their own speciality and capabilities. Now, that's a small overview of what our institute is and what our institute is capable of. Now let's dive into a few projects we had here, just to give you an idea of the kind of projects we're actually doing here. For this project, we were looking at leishmanias, which is a parasite that's transferred mainly due to the bite of a fly. And in this project, we had 3 types of samples. We had a naive mouse, we had a mouse that was infected, and then at a specific time point, euthanized for analysis. And then we had a different time point. And the goal was actually to try and see if there is a specific role of lipids in this disease. So we had liver tissue from naive mouse, infected mouse, and we went and have a look. Can we find some differences in the lipid structure? Is there something changing there? So we could definitely see differences in lipids. We identified numbers of lipids, and already on the ion images, we could see that there was a difference between the infected mice compared to the non-infected mice. If we then do some statistics, we were nicely able to separate the groups. And using that PCA analysis, we can have a look which compounds, which features, which M over Z values, masses, analytes, make sure that these groups are separated. When we then look at the loading scores, we can find some lipids that are very specific to the diseased tissue. If we then correlate it with some regular H&E stainings, we can find some lipids that are only located in the granulomas, so the infected regions of the liver. And if we then do some further pathway analysis, we can hypothesize and find out more how this disease evolves in an infectious state. Very often, people say multi-imaging, it's really nice, you get a lot of molecular information, but you can never quantify using multi-MSI. That's why in the last couple of years, we've been looking more into quantitative imaging. And this was a first study where we looked at quantitative lipid imaging. So again, very similar as to our previous workflow. What we do is we make sections of our tissue, but instead of immediately applying the matrix, what we'll do is first we'll apply an internal standard mixture. So very similar to what we do with LCMS or any other analytical chemistry field. We have internal standards in there to help quantify our lipids. We go to the data analysis, and then we actually get quantities of lipids in our tissue. This was a multi-institutional study, so we tested it in Maastricht, but also in Australia and Wollongong. And we actually came to the following results. First, we optimized the internal standard mixture. We went and have a look at the endogenous signal of all the PE lipids, for example, which is a subclass of lipids. And we tried to find a concentration of the internal standard that's somewhere mid-range. So the internal standard will have the almost the exact same ionization rate efficiency as the lipids that are endogenous to the tissue. We did this for an entire range of lipid classes. And like that, we try to make an internal standard mixture that's actually representing the entire brain lipidome. These internal standards were deuterated forms, so we kind of modified the molecular structure, or there were similar lipids but odd chain. And the benefit of this internal standard mixture is that we saw subtle changes in spatial localization. So if you look at the left bottom corner, what you see is it looks like this and like that. But if we use our internal standard for normalization, suddenly the pattern changed. Here we saw that there are like hotspots all the way in the brainstem. Similar here, we see that the hotspots kind of changed. So there were subtle changes that suddenly became visible that were not visible before. And using this internal standard mixture, what we did was we actually created a quantitative brain atlas. So for different anatomical regions, we determined what's the quantity of lipids that we find there. And like that, we made an atlas of, let's say, a healthy brain. We did that for a number of amounts of lipids. And in the end, we'll use this lipid atlas to compare to diseased tissue. And then I very often get the questions, does this work for all instruments? Because I have a different instrument. So we compared two different instruments, one in Australia, one in Maastricht. The only difference was that the one in Australia was a lot slower, but it was better at determining the accurate weight of the molecule, while ours was faster. And when we look at the correlation plots, there was a very good correlation, except for a few outliers. Good thing is these outliers could all be explained by the fact that in our fast instrument in Maastricht, we were not able to really accurately determine the mass, and there might be some overlap with other molecules, while in Australia, they were able to do that. So how does this compare to regular LCMS methods? If we look at literature, the amounts we found were very similar. And if we compare our data to LCMS data of the same tissue sections, then we found that there was a really, really nice correlation. So our results were very similar in quantity to the amounts that we found in the golden standard, as we call it, LCMS. A quick example of how we can use this. This was a project funded by Michael J. Fox Foundation, which is the guy you all know from Back to the Future. Sadly, he also has Parkinson's disease, so he made a foundation to support research to help him fight this disease. Causes of Parkinson can be a little bit of everything. Infections, metal poisoning, just getting old, sadly. But 5 to 10% is due to genetics. And one of those genes is a GB1 gene, which encodes for GCase. And the goal of that GCase is actually, we have these lipids, and in a normal human, or mice in this case, it cuts the glucose in the metabolism of the ceramides, and we end up with a glucose and a ceramide. Now we see in Parkinson's disease that this GCA is mutated and not working anymore. So we hypothesized that this might play a major role in Parkinson's disease. For this, we needed a new technique, which has now been available, I think, for 3 years, and it's called MALDI-2. So MALDI-2, it already gives away a little bit. We're going to use a secondary laser. On the left side, you see our tissue section being hit by a laser. The ions that are created that would normally go to our mass spectrometry mass spectrometer now suddenly get hit by a secondary laser. We don't know the exact mechanism yet, but what we see is an increased sensitivity for specific molecules. So on the left side, MALDI1, right side, MALDI2, and even some molecules that were not that visible before, like for example, cholesterol, suddenly become visible and can be studied. So we applied this to a mouse model with an increased mutation severity, so heterozygous versus homozygous. And this also represents an increasing risk of Parkinson's disease. And we went and have a look at those lipids that we discussed earlier that we hypothesized that should be there should be an influence on. Now, if we look at the ion images, on the top, we see our wild types or our naive mouse, and then we see an increasing mutation severity and an increasing Parkinson's disease severity. And at first sight, it might not look like a lot is changing, but what we see is that we have these hotspots here, the brainstem, and that's also where we see if Parkinson's disease starts, where our first plaques and our first symptoms appear. So we see an increasing amount because we use the splash mix. We can also quantify the change, and we see an increased amount of these hexosyl ceramides appearing when we increase the mutation. If we now go register this with a regular H&E staining, we can see that there are specific anatomical locations linked to these hotspots. Now, this is just by eye, but we can also do this with statistics, and we do this with segmentation. And what we'll do is we'll look at the data and we'll try to pull them apart. And we do this with a clustering algorithm where we first try to divide it into two groups. Then one of those groups we're going to divide into another two groups. And when we keep doing that, we can do that on our data, and it looks like this. So what we saw was that this blue color really nicely correlates with this lipids and thus places of accumulation. So we identify hotspots for this accumulation, which in the next step, we can section away and actually characterize even better and more accurate compared to regular LCMS, for example, where we very often homogenize our tissue. And as a result, the influence of these hotspots is averaged out. So we don't get that clear results. And that brings us to our next topic of the day. What we also try to do here is combining imaging and omics. We call it multi-omics, and it's a growing field. You see it everywhere. If you write a grant nowadays, everybody wants you to include multi-omics and combining as much modalities as possible. So that's also what we're doing here. And as an example, what we do is we have our tissue and we make sections and we put them on a special slide. It's called a pen membrane slide. And then we're going to do imaging. So we throw it in our imaging mass spectrometer. And when we look at these ion images, sometimes we see similar hotspots as we saw in the Parkinson's disease project. We do the segmentation like we did before, and then we export these coordinates into our laser microdissector. And this laser microdissector has the capability of sectioning out only this small part. And as a result, we can further process it using regular LCMS analysis. So we create results that are very specific to these regions of interest, which is in this case a tumor, if I'm correct. So we use our mass spec imaging to create hotspots that then can be further analyzed using more conventional techniques that are already well established in the research community. We can also afterwards do a staining and then correlate all the data that we have together. If we look at the workflow in different steps, here we see our MALDI MSI imaging with the mass spectrum and then the peaks that we selected. And we clearly saw a region that we were interested in. We clustered it using some statistical algorithm that we discussed earlier. We do the laser microdissection, capture it into Eppendorf tubes, and then we do standard proteomics analysis, followed by a pathway analysis. And like that, we can draw conclusions only from that specific region and look at the proteome of that specific region and look which pathways are influenced there and try to correlate it back to the disease. Now, a new thing in our fields, it's already, I think, a year ago, it went to the market. It's called MultiIHC. So in pathology, we have our regular immunohistochemistry stainings. But, well, this is a next step in that. On the top left, we have our untargeted multi-MSI. So we have our tissue section. We do our regular multi-MSI measurement. We create these ion images using the molecules in an untargeted way. So we don't know what we're looking for. In the next step, we're going to do a staining using photocleavable Maastricht antibodies. So this is very specific antibody to a specific protein that we think will be important in our tissue. And on these antibodies, we also have a fluorescence stack. So we can also create a fluorescence image. And in the next step, what we're going to do is we're going to irradiate these photocleavable mass stacks, and we're going to cut away this linker. This linker has been cut away, and this mass stack gets available. When we then do multi-imaging, we can get a really clear spectrum only from these antibodies. So we create targeted images of these antibodies. Workflow-wise, it looks like this. I'm not going to go much into depth, but this is to break the linker of the photocleavable tags. We apply a matrix. We do our multi-acquisition. And afterwards, when we found regions that we were interested in, we can again put it in the laser microdissector and do proteomics of specific regions. We were the first one to test it for the company. We could nicely see our antibodies back. So some actin antibody for smooth muscle, a HER2, which is a breast cancer marker, vimentin and pan-CK, the keratin marker, and we could create nice ion images of these antibodies. These antibodies then gave us regions that we thought would be interesting. So this is a laser microdissector workflow. First, we have our optical scan. We overlay it with our ion image, which shows us regions that could be interesting. We import it into our laser microdissector, and the laser microdissector sections away only specific parts of the tissue that then will be further processed. And like that, for example, we had a pan-CK marker, antibody marker. We can only section away regions that were very high in pan-CK. Following that, we did proteomics, and we could see the pathways that were influenced, correlated with this pan-CK antibody. We could do the same for all the other antibodies, and in the next step, we could draw results regarding the disease state and the prognosis and so on. Like every field, this is just on tissue, but all the research fields are now moving more and more towards single cells. So of course, we also had to do that. That's how it goes. What you see here is an image made at 10 micrometer spatial resolution. And we selected different lipids that characterize different regions. In the next step, we did an astrocyte immunostaining. We combined it with our MALDI image, and then we could see that there were like specific fingerprints for the astrocytes. This sparked an idea, so that means that our cells might have a specific fingerprint. And that's also a future direction we're moving into. What we're trying to do is actually generate an immune single cell database. So our ideas, every cell has a specific fingerprint. Now, To analyze this, we also need new instruments, but we're also into building new instruments. So we're trying to combine this as a future direction at M4i. But in the end, it's also my opinion, we don't want to do science for science. No, we want to have an impact on society. So we hope that this can be used in the future as a way of doing some digital pathology and as a result get better diagnostics. So that means that we need single cell metabolomic profiles. Already, I think it's already 2 years ago. It was published in 2022, but I think it's 2 years ago. We started with this single cell database. How we do this, we have our microscopic slides. We give it a coding, we wash it, and very simple, we just grow our cells on this microscopic slide. We incubate overnight that it nicely grows over the slide. We do a few washing steps because in the medium, there's a lot that we actually don't want in our mass spectrometer. So we do some washing steps and we end up with a single slide full of cells that can be analyzed using multi-mass spectrometry. We did this for various amount of sites, fibroblasts, macrophages, cardiomyocyte, breast cancer cells, and we actually got some really good molecular profiles. If we ask the software, is it possible to differentiate these cells based on the data we're giving you? On the left top corner, you see in our PCA analysis that it was relatively easy to separate these cells. These are actually 4 types of breast cancer cells, so they're very similar, and even those cells we were able to differentiate. When we look at the loading plots, we can even find some metabolites that are actually really driving this separation. Now, the next question was, can we do this on all instruments? We tested that, and the profile and the patterns were very robust. Independent of sensitivity resolution, our patterns were very robust for these different cells. Then we took it a step further. What happens if we have the same cells, but we change only one receptor on that cell? So here we took different breast cancer cells, one that were positive for an estrogen receptor, one that was positive for a progesterone receptor, and one that was HER2 or HER2 negative. This is very important when we go to the clinic because these receptors really have an influence on the decision making regarding treatment and also the prognosis of the patient. So if we could separate this, this would be a very big step forward. And if you look at the model that we made, you can see that we nicely separate the 3 types of cells, even though they're only different in one receptor on the outer surface. Of course, we want to apply this to actual tissues. So we went and have a look. We have these tissue sections. We asked the pathologist, could you please annotate them for us? And then we went and have a look. Can we do the same using our mass spec imaging data? And the answer was very straightforward. Yes, we could. So we could differentiate tumor region, necrosis region from the normal brain regions. All this data we put into a computer model, a single cell molecular recognition model, very fancy term, but AI is everything nowadays. So we put it into the computer and ask the computer, make us a model to predict if a cell is one specific cell or is it another cell. Try and find a recognition model. We applied that in practice, and what we see here is actually a breast cancer tissue section. And on the left side, you see our laser moving. So it's shooting, and every time it shoots, it gives a prediction of which type of cell it is. Now it's in the hole of the tissue. You can see the laser is in the hole. The moment it starts hitting the tissue, it's going to give you an identity of the cell it's actually hitting. So now we're hitting a breast cancer cell, and the accuracy of the prediction is 93, 95% accurate. In the future, we hope that this will be used in the clinic and will help the life of the pathologist a little bit easier because this is an objective way of classifying tissue into tumor tissue, necrotic tissue, or healthy tissue. Every day we're pushing our science a little bit forward, and at the moment we're even looking at subcellular spatial distribution. Like I said, MALDI nowadays is limited to 1 to 2 micrometers in spatial resolution. That means we can also look inside the cell. And as an example here, we see our staining with the nucleus and the surrounding cytoplasm, and we could find the same back when we look at the lipid distribution, that it's very abundant in the nucleus and less abundant in the surrounding. And different lipids have different distribution, which might help in the end in figuring out the pathology of a disease and so on. So in conclusion, what we try to do here at M4i, and relatively succeeding at, is we're trying to combine different modalities. So we try to do imaging, mass spec imaging, fluorescent imaging, microscopic imaging. We try to couple it to omics, proteomics, lipidomics, metabolomics. With those results, we have a look at the pathways that are involved in specific regions. We link it to the context that we got from the pathologist or from the disease, and then try to link it back to all the things we found. So all those interactions hopefully will help us moving towards a better understanding of different diseases and a more precise way of treating all these diseases. And with that, I would like to end. This was not only my work, but we have a very big group and a lot of people participated in these research projects, for which I'd like to thank them. And of course, we also get a lot of funding from different institutes. So that was it. If you have any questions, always welcome to reach out, or you can just ask them in the Q&A that's